Main objectives of this lecture is to understand about milk flora and types of microflora. So first, milk flora. Milk, when secreted in udder by the mammal, it is free from microorganisms. That is, it is sterile milk. During the production, handling, transport, and during the distribution of milk, microorganisms enters in the milk and contaminate that milk. Microorganism entering in milk may be pathogenic or non-pathogenic to human being. Growth of these microorganisms in milk affect the quality of milk by bringing about spoilage of milk. Rate of spoilage, which is caused by microorganism, will depend upon the number of microorganisms present in milk. It will also depend upon the condition under which milk is stored, and it will depend upon the length of time of storage of that milk. The number and types of microorganisms present in raw milk will depend on nature and extent of contamination of milk, which varies with the condition of milk production and subsequent storage of that milk. Production of milk under hygienic condition and subsequent storage at low temperature, that is less than 4 degrees Celsius, restrict contamination and proliferation of microorganisms in milk. Microbial number. The total microbial load of raw milk can be determined by standard plate count. Bacteriological standards for raw milk, which is recommended by ISI, Indian Standard Institution. Depending upon the number of microorganisms present in milk, milk can be graded in 4 qualities. If milk contains the microorganism not exceeding 2 lakh SPC, standard plate count, per ml, then that milk is of very good quality. If number of microorganisms present in milk lies in between 2 lakh to 1 million, then milk is of good quality. If number lies in between 1 million to 5 million, then fair quality milk. And if this number is more than 5 million, then that milk is of poor quality. Types of microflora. The heterogeneous population of mesophilic, thermoduric, thermophilic, psychrotrophic, and pathogenic organisms constitute the microflora of raw milk. So first, mesophilic microflora. Mesophilic microflora includes microorganisms which grow optimally at a temperature in between 30 degree to 32 degree Celsius. They are called as a mesophilic organisms, which includes different species of micrococci, that is Micrococcus flavum, Micrococcus varians, Micrococcus caseolyticus, Micrococcus fuderentia, Micrococcus luteus, and Micrococcus roseus. Staphylococcus aureus, Staphylococcus epidermis, Streptococcus agalacti, Streptococcus uberis, Streptococcus zymogens, Streptococcus lactis, and Streptococcus faecalis. Asporogenous gram-positive rods, that is non-spore-bearing gram-positive rods, which includes Corynebacterium diphtheri, Corynebacterium pyogenes, and Corynebacterium bovis. Species of Mycobacterium, which includes Mycobacterium tuberculosis and Microbacterium bovis. Lactobacillus casei, Lactobacillus plantarum, Lactobacillus acidophilus, and Arthrobacter species. Spore-bearing gram-positive rods, which includes Bacillus cereus and Bacillus subtilis. Gram-negative rods, including coliforms, includes Pseudomonas species, alkali genes, Escherichia, Enterobacter, Klebsiella, Serratia, Flavobacterium, and Acinetobacter. Miscellaneous mesophilic microorganisms includes actinomycetes and yeast, for example, Candida and Saccharomyces. Molds, that is fungi, includes Aspergillus, Rhizopus, Mucor, Penicillium, and Geotrichum. All these are the examples of mesophilic microflora, that is the microorganisms which grow at a moderate temperature in between 30 to 32 degrees Celsius. Next to mesophilic microflora is thermoduric microflora, which includes the microorganisms which survive at pasteurization condition at a temperature in between 60 degrees Celsius to 80 degrees Celsius, but these microorganisms cannot grow at that temperature. The examples of thermoduric microflora includes the spore-bearing Bacillus and Clostridium species, Micrococcus, alkali gene species, etc. Next to thermoduric microflora is thermophilic microflora, which includes the bacteria which can grow at a higher temperature near about 55 degrees Celsius, and they have an optimum growth temperature in between 45 to 60 degrees Celsius, while maximum growth temperature is up to 70 degrees Celsius. Thermophilic microflora includes aerobic or facultatively anaerobic spore-forming rods. For example, Bacillus styrothermophilus, Bacillus circulans, Bacillus thermoacidodurans, all species of Bacillus, and Lactobacillus thermophilus. Psychrotrophic microflora, which includes the microorganisms which can grow at a commercial refrigeration temperature in between 2 degrees to 7 degrees Celsius, regardless of their optimum growth temperature. The optimum growth temperature of this microorganism may be 15 to 25 degrees Celsius, but this organism can able to grow at refrigeration temperature in between 2 degree to 7 degree Celsius. Psychrotrophic microflora includes gram-negative rods and gram-positive rods or cocci. Gram-negative rods includes Pseudomonas species, Acinetobacter, Flavobacterium, Achromobacter, Alkaligenes and Enterobacter, while gram-positive rods and cocci includes Bacillus, Arthrobacter and Streptococcus species. Coliforms. Pathogenic microorganisms in raw milk. These pathogens, they come in raw milk through different sources, which includes if that mammal is suffering from any disease, then it will act as a source of that particular pathogen. So this mammal secrete this pathogen into the milk and causes the infection to the consumer who consumes that particular milk. Another source is the contamination of milk from infected human handler as well as the fecal and non-fecal sources. The examples of pathogenic microorganisms includes Bacillus cereus, Listeria monocytogenes, Yersinia enteropolitica, Salmonella species, Escherichia coli, Campylobacter jejunum, species of Aspergillus, Fusarium and Penicillium. So milk contains heterogeneous population of mesophilic, thermoduric, thermophilic, psychrotrophic and pathogenic microorganisms. References: Book of Outlines of Dairy Technology by Sukumar Day, Milk and Milk Products by Henry Eccles, and A Comprehensive Dairy Microbiology by J.S. Yadav. Thank you.